be talking about insights into immune biomarkers. And the way we're going to run it, my name is David Rimm, and I'll be the moderator today, but I don't have to do any work other than introductions. The way we're going to run it today is, as was in the program, was that we'll have a discussant speak for about 15 minutes. Then we'll have the 4 poster presenters come up and occupy these seats, and then we'll do questions for about 15 minutes, and we'll have the next discussant. And in order to try to keep on time, we'll start up right away. The first posters, PS 101 to 05, or 04, I guess, will be discussed by Frederique, Penault, Llorca. I'm not so good at non-English words, but Frederique will start us out. Go ahead. Thank you.
Thank you very much. I would like to thank the organizing committee for giving me the opportunity to discuss those 4 very interesting posters on new insights into immune biomarkers, the patient and the subtype of breast cancer. So I'm trying to move my slides. Maybe from okay. So this is my disclosures. So we are going to go through racial disparities and focus on Black and Hispanic patients with poster 1 and poster 2. Then to young patients, and I will focus on the impact of pregnancy. It's poster 4. Then we will go through, in fact, TILs, tertiary lymphoid structure, TLS, and circulating white blood count. Okay. And we are going to look through triple-negative breast cancer in poster 1 and poster 2, HER2-positive breast cancer in poster 3, and young patients in poster 4. And this will all be inside neoadjuvant setting and PCR. So first of all, racial disparities, and I will start with poster 2 from Sharma and colleagues, the impact of racial differences in circulating blood components and S-TILs and outcomes in triple-negative breast cancer. So the context is that triple-negative are overrepresented in Black patients. They have a worse clinical outcome. It can be explained by social disparities, but also by specific biology of triple-negative breast cancer, and probably by specific risk factors. The aim of the study is to investigate combined impact of racial differences, looking at stromal TILs and peripheral white blood cell component on the outcome of triple-negative breast cancer patients treated by neoadjuvant chemotherapy. They have a large series of 469 patients treated by neoadjuvant treatment. 1/3 was treated with Pembro, but the study was not designed to look at the effect of Pembro. They look at several parameters, including body mass index, the circulating blood cell components, S-TILs, and they looked at PCR and RFS. Compared to non-Black patients, the 14% Black patient in this series had higher BMI, S-TILs enrichment, and this was statistically significant. There was no difference in terms of PCR rate with the rest of the population. But there were differences in the white blood count. They had lower absolute neutrophil count and ratio neutrophil lymphocytes. They have higher lymphocyte, absolute lymphocyte count, and lymphocyte monocyte ratio. In Black patients, this low neutrophil count can be explained by Dufinel phenotype, but it's not known to increase lymphocytes counts. The Dufinel phenotype is seen in approximately 2/3 of African Americans. It's an evolutionary trait that's conferring partial resistance to sickle cell disease, and the absolute neutrophil count is 40% lower without conferring an increased risk of infection. It can be a real problem for entering in some clinical trials. And now it's very well studied, and it's well known to drive an increased pro-inflammatory signaling with senescence of the immune system and predisposition to autoimmunity and cancer. When they look at the impact of circulating blood component and S-TILs on PCR and RFS, first of all, RFS was similar in Black and non-Black patients. The S-tills, as I said, were higher, but it did not translate into an higher PCR rate in an RFS in the Black women. And in univariate analysis, we had BMI, absolute myeloid count, absolute lymphocyte count, and LMR that were associated with lower RFS. But in multivariate analysis, it was increasing ALC and LMR that were independently associated with a lower RFS. So in this poster, we are really here with hypothesis generation. Higher lymphocyte count in Black women are probably related to underlying chronic systemic inflammation and higher BMI. There is a weak inverse association of AMC with S-tills, so probably going into tissue with an impact on the immune microenvironment and immunosuppression via TAMS, that should be studied. And if we look at the population, the overall BMI was quite high in this study. It was statistically significantly higher in Black women. But the impact on the white blood cell immune environment seems to be really different in the Black women population. So really, there are a lot of things to go through to look at the impact of the Duffinel phenotype on TNBC outcomes in the Black population. Limitation of the study, small numbers of Black patients altogether, 14% of the population. And we would really like to see the data, specific data on the pembro arm in this population. Now I move to the next poster on TNBCs by Dr. Alberts and colleagues, TILs as a predictive marker of PCR in a diverse patient population with early breast TNBC treated with neoadjuvant Keynote 522 regimen. So it's a retrospective analysis from 184 patient files. All the patients had the Keynote regimen completed, and they had TILs reported. Median age was 51 years old. You can see here that you have a diverse diverse PCR rates. I will go back to that on next slides, but 64% PCR in the Hispanic population. Altogether, 55% of the population achieved PCR. The TILs were clearly associated with an improved PCR rate of 70% in the global population versus 48% without. So the numbers you see here are not percentages on the scales, it's a number of patients. There was no difference in terms of PCR or TILs across different ethnicities. The only significant association were in the Hispanic population. And we can see also that tumor burden matters because when the patient had node positivity, they had higher levels of PCR with pembro. We have recently the update of overall survival in the Keynote 522. And in the supplementary data, you have the OS by ethnic groups and geographic regions. And you can see that there is no significant association with OS in the Hispanic population. So will the Hispanic Latino patient drive an higher benefit from Keynote 522 regimen in real-world evidence data? So from this study, you can see really that the Hispanic population in this group had really higher PCR and an extremely high level of PCR when they had TILs, 80%, and very low level when they didn't have TILs. And we clearly have an association between TILs and highest PCR rate. We had data in this congress on Latin American patients, the PETRA study, with 212 patients and a PCR rate of 59.9% in real life in Latin America with Keynote 522, and another poster on a US population with two institutions where they had 67.3% PCR in the Hispanic group versus 51% in the African American and 63.8% in the non-Hispanic white. We had altogether 25 real-world evidence posters on Keynote 522 that will be presented in this Congress with diverse level of PCR. And in France, we have collected around 8,000 patients with 70% PCR rate. So I would like to insist on the fact that it's very, very important to collect those real-world evidence data. The limitation of the study, small numbers, and TILS data from electronic files, so I don't know exactly the cutoff, and we have binary results, yes or no. So in conclusion for those two posters, response to neoadjuvant chemotherapy plus minus immune checkpoint inhibitors in TNBC from diverse patient population. We clearly have racial disparities, and they are not, those patients are not well represented in clinical trials. So we really need to have further investigation to understand the interaction between race, ethnicities, and the immune microenvironment of the tumor. There is a very important to get TILS data from KEYNOTE-522 trial and aggregating real-world evidence on KEYNOTE-522 regimen efficacy in diverse population with race and also with comorbidities. And I really insist that you look at the different posters on that. And the other biomarkers, circulating white blood cell count and the ratio, and also the very interesting TNBC-DX that will be presented also today with data on not on Pembro, but with other immune checkpoint inhibitors. And altogether, TILS and circulating white cell count and the ratio could be extremely affordable potential biomarker for checkpoint inhibitors. Now we move to the poster number 4 from Dr. Tesch and colleagues looking at TILS and response to neoadjuvant chemotherapy in young patients with breast cancer. So they did an extensive evaluation of TIL subtypes in 131 patients in pre-neoadjuvant chemotherapy biopsy, so no immune checkpoint inhibitors here. They looked at Akoya systems with TIL stromal and tumor compartments, correlation with PCR, grade, stage, subtypes, and germline BRCA and pregnancy. Majority of Caucasian, aggressivity of the cancer, majority of grade 3, majority of HER2 positive and triple negative breast cancer overrepresented in this young population. 13.9% of germline BRCA mutation. The HER2 positive and triple negative had high TILS, and the HER2 positive patient 32% PCR and 26.8% in the triple negative. The major results are that distribution of TIL subtypes varies according recency of pregnancy, germline BRCA1 status very different from germline BRCA2, tumor characteristics, and the increased level of TILS, once again, predict PCR after adjustment, stage, grade, and subtype. When you look at the type and localization of TILS, you have a lot of data, so now we really need to have immunologists digging into the signification of the different subtypes, but major results, TILS are not associated in this study to age, race or ethnicity. They predicted PCR independently of breast cancer subtype, and the tumor TILS, tumor-associated TILS, were more predictive of PCR than the stromal TILS. Looking at pregnancy effect, there were very interesting data at diagnosis with very low levels of TILS in the population of pregnancy, cancer during pregnancy, with also a higher population after tumor after 5 years of pregnancy, different from nulliparous women. So really, I think those results are interesting and they need further exploration on the effect of pregnancy on tumor microenvironment and after. Limitation of the studies. For me, you need to explore beyond T-cell markers, and there are some missing data on the pregnancy that should be gathered to have more explanations. So in conclusion for this poster, young age, we have the importance of specific exploration, looking at TILS, proportion, location, and subtypes that are associated to response to neoadjuvant chemotherapy without checkpoint inhibitors in those young patients. We need specific exploration of intrinsic biology, the influence of pregnancy on tissue microenvironment and the immune tissue environment via tissue remodeling and immunosuppressive effect to understand pregnancy-associated breast cancer. And we need to explore beyond T-cell, looking at B-cell, TLS, and macrophages. And this brings me to the last poster by Dr. Zhang and colleagues, prediction of prognosis and efficacy of neoadjuvant therapy in HER2-positive breast cancer patient based on tertiary lymphoid structure. The tertiary lymphoid structure are secondary lymph node structure in tissue. They are critical regulators of the anti-tumor immune response, and they are able to recreate an intratumoral immune cell cycle. They are associated with improved prognosis in several tumor types, and they are associated to response to immune checkpoints inhibitors. So in this study, they did 2 in silico studies from Metabric and I-SPY trial HER2 positive, and they looked at some biopsies from their files. They used 3 different approaches to evaluate the TLS. So in the Metabric cohort, they use a 9-gene TLS gene set. They confirmed the prognostic value of the TLS in this HER2-positive population. They had 23% TLS-I in the Metabric with a stronger immune response on CyberSort, and the low TLS population had an increased proliferation activity. In the I-SPY2 trial, TLS were predictive, highly predictive of PCR. You can see here. ITLS were associated with the higher PCR rates, but the addition of pertuzumab increased PCR rate in TLS low. And in their biopsies, in 19 cases, they determined TLS by multiplex immunofluorescence, and they showed also a very high correlation with PCR, but they had half of their biopsy with high TLS. So the study limitation here is different approaches to measure TLS, showing the lack of standardization. So the conclusion for this poster is that TLS in HER2-positive disease are prognosis and predictive factors. They need to be studied in HER2-positive, but also in triple-negative breast cancer, and they need further investigation because they can be predictors of response and allow treatment adjustment. But there is an absence of internationally accepted criteria on how to define TLS using histopathological slides, spatial proteomics, spatial deconvolution, immunohistochemistry, or molecular signatures. So really, we need standardization before translating to the clinic. To summarize, we need more diversity in clinical trials, gather large real-world data, and collect white blood cells information, especially in the KEYNOTE-522 context. TILs matter, and they are here to stay, and I really encourage you to listen to Roberto Salgado this afternoon. We need to have their status in KEYNOTE-522. It's very important, I think, to have the TIL status there, and they should be added in practice guidelines. A young patient, we need to look at the impact of pregnancy and the importance of exploring beyond T cells, and for TLS, they are promising biomarkers in HER2-positive breast cancer. But standardization is required. I'm sorry, I've been a little longer, but thank you.
So what we're going to do is, maybe I can get the microphone to work here. If I can ask all the poster representatives to come up, and then we'll have a few minutes of questions. You can put, if they can come up and occupy these chairs. If you were a poster presenter, I think we should have 4 of you here. And then while that's happening, if you have questions, you can ask questions either by going to the microphones. There's 2 microphones in the room, or you can put them into the app, and then I can see them up here and I can ask them. So we're having trouble getting our poster presenters. Come on up. I'm not sure who's. We should have 4 chairs here. Do you want to move over? And let's see. We have a question at the microphone to start us off. Go ahead. Just introduce yourself first and say your institution, and then go ahead and ask your question to the person that you'd like to discuss.
Okay, thank you. I will keep the introduction very short. I'm Yale pathologist from the Netherlands Cancer Institute in Amsterdam. Very interesting results. I was wondering, for the racial disparities results, to what extent larger tumors may lead to another, I would say, immune response around these tumors, because it's a dynamic thing. yeah, the ones that were involved in these racial disparities posters. Maybe they can answer these questions.
So is this a question for Riya or Dr. Albert?
Yeah.
Go ahead.
Yeah, I think that's a really interesting question, especially considering in our study we did find that once there was nodal involvement, patients in the subgroup had higher rates of PCR when TILs were involved compared to patients with nodal involvement without TILs involved. So I think that's something that could definitely be explored further to see like larger tumor burdens, more nodal involvement, see whether there's more stratification there.
Thank you.
Okay, then don't be shy to come up to the microphones, but someone put a question in that I'll ask each of the investigators that are sitting here, and that is, what is your definition of TILs? That is, the TILs can be defined in different ways, and one of the things that Dr. Penault Llorca said is that, it's important to standardize. And so if each of you can answer the question, maybe starting with you, Riya, how did you define TILs for your study, and how does that fit in with standard definitions from the TIL working group?
So at UT Southwestern, the pathologists, they report TILs manually and visually, and they normally include the presence of TILs when it's quantified as 30% or higher. So that was our cutoff for including TILs in our study.
So that you use the international working group criteria, but then you use a 30% cut point.
Yes.
Okay, let's see. Dr. Tesch? Yeah, there you go.
Our study, we used like multiplex immunofluorescence, so we were using sort of more novel methods. And in that study, given there is no sort of recommended cut points, obviously, when you're using that kind of technology, we were defining TILs. We were looking at it in association with the clinical pathologic characteristics. We used the median as the cut point for high versus low. And then in the multivariable analysis, it was looking at it by 10% increase in expression.
So did you compare that to pathologist red TILs or just use the IF?
So we do have some data on lymphocytic infiltration that was done sort of predating the recommendations from the TILs working group. And we are looking at that in further detail, just correlating it with the multiplex data. Yeah.
Dr. Zhang?
So thank you so much. So thank you, the Professor Frederique, giving me the very important suggestion to how to define the TILs. So this TILs is not like the TILS. It's just like the lymph nodes in the tumor tissue. So we just follow the picture that's published on Nature. They use 9 gene to the expression to define the TILs. And they also found this will be improved immunotherapy in the melanoma. So we just wonder if this TILs will infect the breast cancer. So we use a gene expression we tested in the public data, just like the METBRIC and the TCGA. And we still need to do some more work to find if in the HE slide or we use the IHC slide to find the more confirmed structure in the breast cancer.
So by using the gene expression
Yes.
as a TIL definition, how did you find a cut point?
We use the let me see, RES dot point cut, the R package, the Suverminer. So this defined in this paper. So we just
So it was an optimal cut point. It was not a predefined, prescribed.
maybe
cut
the 0.32.
So, okay, thanks. Go ahead.
I don't know if Dr. Sharma is here for a poster. Okay,
Oh, Dr. Sharma is participating by Zoom, but we can't hear her.
we can hear you.
I'm not sure if she's on mute or if you... There we go.
Okay.
Yes, can you hear me?
Now we can hear you, Priyanka. Go ahead and what was your definition of TILs?
So we used the international working group criteria with pathologist red TILs, and they were reported in increments of 10. So it was kind of continuous we use a 30% cutoff to define, two categories because that cutoff has been used before in previous studies and seems to discriminate well in terms of outcomes. But we do have continuous reads at every 10% category.
Okay, thank you. I encourage everyone to come to the mic. We have time for one or two more questions, and there's another question on here. In defining Black or Hispanic populations, were the populations in each case self-identified, or were there other methods for defining the ethnicity? I guess maybe you can go first, Priyanka.
It was self-identified.
Self-identified, okay.
So our demographic data was also collected from chart review, from patient charts, so it was also self-identified.
But from chart review, okay. Okay, and we have time for one more question, so I'll take the moderator's prerogative and ask the question. My question is for Dr. Zhang. In the case of tertiary lymphoid structures, how did you compare them to tertiary lymphoid structures? One of the questions that we ask as a pathologist is our biopsies are so small we can't necessarily see a tertiary lymphoid structure in a biopsy specimen, even though we can often see them in resections. How did you address that issue, or were your gene expression tertiary lymphoid structures all from resection specimens?
We use the patient in our department to diagnose the HER2 positive. We just use the slide to use the multiple IF to find the structure, but we have the number of the patients is too small. It's only 19 patients, so we need to explain this data.
Okay.
So we still want to use the HE slide to find confirm the data to support that.
So it's small data set and need to confirm with H&E. Okay. Oh, there's another question here. You want to read it? I can't read, and I think we have to change it around.
No
next time
worries.
and have the-
Yeah, I just noticed there was another question that did lower ANC result in lower RDI? I think this may be for Dr. Sharma.
Dr. Sharma, did you hear the question?
In general, the chemotherapy, preoperative chemotherapy delivery rate was similar across blacks and non-blacks.
It was not the question, no?
Okay, I think that since we're at 8 o'clock, we're going to stop at this point, and I'll ask these speakers to step down. And then next, we're going to have a discussion of the next 4 speakers, and we'll do it the same way. Dr. Theo Foukakis will present a summary, and then we'll have 15 minutes for questions again. Thanks to the speakers. So you can come up here. Here's the slide.
Let me see. And advance with this, right? Yeah. Okay. Good morning, and I would also like to thank the scientific committee for inviting me to discuss these very interesting abstracts and provide the oncologist perspective in those. And let's see. It doesn't advance. Oh, no. Okay, here is my disclosures. I'll do it manually. And these are the 4 abstracts that I will discuss today, and start with the first one, genomic characteristics related to histology-based immune features in breast cancer by Dr. Zhang. And to put this in context, I think we have to consider this publication, cardinal publication, published a few years ago by Dr. Carsten Denkert and the colleagues at the German Breast Group. In this study, they took biopsies from more than 3,000 patients treated with neoadjuvant chemotherapy in 6 different trials in the German Breast Group, and evaluated TILs in this. And what they could see here is that TILs could predict predict PCR in all clinically defined subtypes of breast cancer, with high TILs being correlated to high PCR rates. And in addition, they showed that TILs were prognostic beyond PCR for long-term outcomes, both in terms of disease-free survival and overall survival. But interestingly here, this was seen only for non-luminal subtypes of breast cancer, while in the ER-positive, HER2-negative disease, the correlation was inverse, saying that high TILs were associated with worse overall survival. And going back to Dr. Jin's poster, what the researchers did here was that they used the TCGA and did a pathology review of the immune infiltrate, including TILs and TLSs, and they generated the gene expression signatures based on these features. And then they used these signatures and analyzed their score their expression in different PAM50 based subtypes in the TCGA, and only in focusing on tumors with more than 10% of stromal TILs. And then they validated the results and showed their prognostic, the prognostic significance of these signatures in the scan B cohort. The results, they show that the immune-related histologic features are abundant in HER2-enriched and basal-like PAM50-based subtypes as expected, and they also showed that there were distinct signatures in the different PAM50 subtypes, as you see here in the right panel, where the luminal A tumors were associated with mast cells signature. In luminal B tumors, there was a high score of the interferon signaling. In HER2-enriched, the B cells, and in basal-like T cells signature. And finally, they could show in the scan B cohort that the signatures were prognostic in non-luminal subtypes as expected. So in summary, Dr. Jin colleagues, they showed that the immune infiltrate is heterogeneous, both in terms of biology and clinical significance in the breast cancer subtypes, and they're also distinct, at least when it comes to the cases with high TILs. And questions remain: is bulk gene expression analysis better than using histology that is used in most of the studies? And the one obvious caveat in the gene expression signatures is, of course, that the spatial heterogeneity is not addressed, or at least partially, with the methodology that the researchers used here. And of course, an obvious step would be to compare these signatures with stromal TILs or AI-based TILs in the scan B cohort. And of course, the prognostic and predictive implications are not fully addressed, as the scan B is a population-based cohort. The patients were treated according to routine, so we cannot be sure what the predictive information of these signatures may be. And I'll move to the second abstract by Dr. Fukumura-Koga. And in this study, the researchers used a cohort of 84 triple-negative breast cancers treated with primary surgery, and they used immunohistochemistry to stain for CD3 and CD8. And then they used both a manual and an AI-based evaluation of the numbers and the spatial patterns of TILs. And as you see here on the right panel, the method that they used included a definition of the central versus the peripheral stroma using the invasive margin of the tumors, and they could then see the abundance of the immune cells in the different compartments. And what this analysis showed, first, the manual enumeration of CD8 positive cells was associated with a better prognosis in these tumors, and the infiltrative pattern, the eye here with both high abundance of TILs both in the central and the peripheral stroma was also associated with better prognosis. Of course, producing special immune infiltration metrics is not totally novel, and I would like to show this very interesting publication by Dr. Feng and colleagues that came out a couple of years ago in Nature.com, and the researchers here produced two different metrics, the normalized mixing score and the gradient entropy slope, both of them essentially showing us whether the immune cells infiltrate the tumor or are separated than the tumor. So with a high score showing a high mixing of the tumor cells and the immune cells, leading to higher PCR rates, and similarly when an attraction entropy is seen in the tumor, then it's also associated with higher PCR rates as compared to the repulsion pattern of entropy. So going back to the study by Dr. Fukumura-Koga, this is a relatively small cohort, and the patients are treated with primary surgery, which is not the standard practice in triple-negative disease today. And they also used immunohistochemistry, which is not as common as the evaluation in the standard H&D sections that we use are used in most AI-based algorithms today. And of course, the question remains whether this can be applied in core biopsies that are used in triple-negative for neoadjuvant therapy, as the invasive margin cannot really be defined in the biopsies. Then we move further to the third abstract, which Dr. Pan and colleagues at MD Anderson used the Artemis trial of neoadjuvant chemotherapy in triple-negative breast cancer, including around 400 patients, and they used an AI-derived TILS evaluation that was compared to manual TILS enumeration and manual Ki-67 assessment in these tumors, and compare their prognostic significance. What they showed is that the AI TILs were moderately correlated with manual TILs, and they showed that the AI algorithm had the potential to enhance PCR prediction when added to manual TILs and Ki-67. However, this was not obvious in the validation part of the Artemis cohort, where the AUC was not improved by the addition of the AI TILs to standard enumeration. And of course, the AI-derived TILs has been addressed before with pioneering work from Professor Rimm's group shown here, in which a cell classifier was produced using H&E slides and defining tumor cells, TILs, stromal cells, and other cell types. And based on this classifier, 5 different TILs metrics were produced. I'm not going into detail on them, just showing that the 5th one, EAS TILs, more or less corresponds to the TILS evaluation proposed by the International TILS Working Group. Both in the initial publication and then in our hands, this TILS algorithm has shown to have a high concordance between the digital and the manual TILs, both in surgical specimens and in core biopsies, and then a bit lower when using TMAs. The difference in the methodology by Dr. Pan and al. Is shown here. They use the tissue segmentation model, which offers the advantage that no manual annotations are needed. And in addition, the cell classification correction is based on the tissue classification, which is a very interesting approach in order to improve the cell class predictions. That is a major question in these models. It is unclear from the poster whether this algorithm was previously validated or was trained in the Artemis cohort, and whether it has been fine-tuned for breast cancer, as the initial AI pipeline was optimized for lung cancer, as shown in the reference used in the poster also. But the question, of course, remains here, does it matter which AI-based algorithm we use, or can we use them interchangeably? And this question was addressed by Balázs Ats, pathologist at Karolinska and his group, where they took about 10 different AI algorithms to score TILs in triple-negative breast cancer, and they evaluated the analytical validity and their clinical validity in scan B. What was shown in this paper that was published just a couple of weeks ago is that there was a high correlation between the different algorithms, and they were also highly correlated to the manual TILs shown in the last column here to the right, with coefficients of about 0.7, which is higher than the one shown in the paper here. And also the clinical validity was demonstrated with quite consistent results among the subtypes. In summary, this paper shows the results of an AI TILs methodology that is very interesting. Questions remain whether it's been validated and how it correlates with standard manual TILs evaluation, and also how it performs not when added to manual TILs, but when it is used instead of TILs. And of course, Artemis is a fantastic trial, but is using only neoadjuvant chemotherapy without immunotherapy. And it will be, of course, interesting to see how this and other algorithms perform in Keynote 522 and other trials using neoadjuvant immunotherapy. Of course, in addition to the algorithms evaluating the stroma, there are other AI-based algorithms, for example, using digital Ki67 or digital grade that could be added and see whether they add value to these classifiers. And going to the last abstract by Dr. Marcela Carausu in Christos Soteriou group in Brussels. Here, the researchers here, they used single-cell RNA sequencing and single-cell T-cell receptor sequencing of biopsies collected longitudinally in patients treated with neoadjuvant therapy for breast cancer. All subtypes were included, and biopsies were taken both at baseline and during treatment. In summary, the results here show that neoadjuvant therapy leads to a decrease in T cells, B cells, and plasma cells, and an increase in fibroblasts. And specifically in patients responding to treatment, there is an increase in the CD8 to CD4 Tregs and a decrease in naive and early activated T cells. And interestingly, the subtype of cell clone of CD8 positive CXCL13 was positive cells was expanded in triple negative breast cancer responders. And we addressed the question of single-cell sequencing in the longitudinal biopsies a few years ago in this paper with Nick Navin and my group at Karolinska and MD Anderson, where we took longitudinal biopsies from patients treated with neoadjuvant chemotherapy for triple-negative breast cancer. And we could show here that in resistant cases, there was a selection of resistant clones after treatment, but also a transcriptional reprogramming of the cells leading to the resistant phenotypes. And this study was focused on the epithelial compartment of the tumor, and I think it's important to consider this also in the current and the other studies, also separately, but also when relating to the stromal, the tumor stroma studies as shown here. And I would like to just stay a bit more on the CXCL13 positive cells that were shown in several publications now that the CXCL13 subtype can be a good biomarker of tumor-specific CD8 and CD4 positive cells as opposed to bystander T cells. And this has been shown in several single cell cohorts, single sequence cohorts, including breast cancer cohorts, that it is correlated with favorable responses to immunotherapy, and the subtype is also increased in cells responding to therapy. So it's a very interesting marker that I think worth further consideration. The T-cell receptor sequencing shown here is very interesting, but as with other single cell assays, here we miss the spatial component, and I think that this is a question that can be addressed by spatial transcriptomics, and this is a method that the spatial VDJ that was published by Camilla Engblom and colleagues a year ago in Science, showing that special annotation of full-length T-cell antigen receptor transcripts is possible, so providing both whole transcriptome and the tissue morphology at the same time. And it is a very promising method for high fidelity mapping of the special and the special lineage tracing of the B and T cell clones. So I'll summarize here the four abstracts. They show that the immune features are distinct among the different PAM50 subtypes, and the AI-based spatial analysis of TILs is promising with potential prognostic implications in triple-negative breast cancer especially. And finally, that the single-cell RNA sequencing of tumors on treatment identified immune populations correlated with response to treatment. It is worth of further consideration. And finally, I would like to thank the authors for sharing their posters and my research group members for their comments on the papers. Thank you for your attention.
Okay, excuse me. Can we ask the speakers, the poster presenters to come up? And in the meantime, a question is posed as a patient advocate. I don't know the answer to whether oncologists always measure TILs or whether they just don't tell the patient. And for the patient advocates in the room, I encourage you to attend, and actually everyone in the room, I encourage you to attend the session on TILs at 2 o'clock this afternoon, when you can get an answer to that question. But in general, TILs are not standard of care in the US, and they aren't done by the oncologist, they're done by the pathologist who's reading the slides and conveys that information to the oncologist in general. For more details, I would encourage you to come to Dr. Salgado's talk at 2 o'clock to sort of hear about the history of TILs and how they're actually done and whether or not they should be used on every patient, which I think Roberto would say yes. But for the advocates in the room, that's not the current standard of care. Okay, I don't see any. Let's see, we're all set here with the poster presenters. I don't see anyone at the microphones. And maybe I'll start with the question while you all think of questions to ask. And ask, let's see, it was the difference between I-TILs and S-TILs was done in the poster with using the Halo technology. I think this was Dr. I'm trouble finding it here. Dr. Pan? No. Who was the, okay. Can you clarify how you used, how you defined I-TILs versus S-TILs? Oh no, sorry, it was.
I'm not good at.
It's Dr. Makiko Fukumura-Koga.
Yes, I'm not good at English. I'm sorry. easier question is how I do.
So what is the difference? How did you determine, because you talked about I-TILs versus S-TILs, and there is a standard way to define that from the International Working Group, but because you used HALO to define them, how does that compare to the standard from the International Working Group for I-TILs, for definition of I-TILs versus S-TILs?
We evaluated the I-TILs hard and more circumstances connected to malignant tissues is I-TILs, and less than half circumstances connected to malignant tissues detected S-TILs.
Okay, so hard to understand. Maybe a hard question, but I think we'll move on. We have a question at the microphone.
So Harry Baer from Virginia Commonwealth University and Massey Cancer Center. I'm standing here as someone who does mouse and human immunology in breast cancer and admitting to be confused because in a lot of models, including adoptive therapy and mouse models and CAR T cells, the effort is to prevent T cell exhaustion because exhaustion is considered to be bad or counterproductive. And yet we're seeing a lot of the single cell RNA-seq and other data saying that the presence of exhausted T cells predicts for a good response. So I'm admittedly confused.
Okay, do you want to take that or should I?
I can, yes. Yes, indeed, historically, this is what we know, that exhaustion is bad. But yes, I think as we discussed before, there is also a continuum in the states of exhaustion. And at least some markers that we consider as markers of exhaustion, as CXCL13, are also markers of tumor-specific response. So maybe, yes, and these studies with single-cell RNA-seq induce us that maybe we should reconsider the idea that we have on exhaustion, that it's not always just the bad thing, and these markers can also mean that there was a chronic activity before. So they were initially tumor reactive, and also there are more and more studies that show that they have another role in the formation of TLSs, these exhausted T cells. So yes, I think they're interested to do these studies because they show us another way to look at also at these exhausted T cells.
Does that answer your question? Okay. So are there any other questions? If not, maybe I'll ask another question to Dr. Pan. You looked at both Ki67 and TILs, and was there an effort made to try to combine those into a single parameter or a single model, or can you clarify how that was done?
Yes, in our work, we do incorporate the three factors. One is the Ki67, the other two is from manual TIL, and the third is the AITL. So the main aim is that we have seen that the AITL and the STL, they have a moderate correlation. So in that context, we hypothesized that the AITL could be complementing to manual TIL. So that's why we're incorporating the three scores. Regarding the Ki67, we basically follow a prior work where they show that Ki67 is an important biomarker for predicting PCR.
So, but I guess the question is, have you tried to combine them, or you just use them individually? And each
combine them, yes.
Combine them, okay. Do you have any questions? Let's see if I can add to Dr. Carausu another question about, have you ever compared the genomic TILs to this, or tried to determine this comparisons with spatial TILs to determine if that's really, is the genomic signature you have to get from grinding up, right?
Yeah, for now we didn't do special studies on this slides. This is the next step, indeed.
So you don't know if they're from iTILs or STILs. It's just the whole tissue.
Yes, for now is single cell, like bulk, indeed. But indeed, the next step would be to look at their spatial organization, also to look on the TILs, like histology, pathological TILs, and also do some more in detail spatial studies, either with multiplex EF, IMC, or spatial transcriptomics.
Okay, Dr. Jin Cha, do you want to comment on that? Because you also talked about genomic methods for TILs.
Actually, my family name is Cha. So.
Cha, okay, sorry. Dr. Cha? The question is, have you correlated the genomic signatures that you worked with any spatial information, or how do you distinguish the origin of the genomic information?
In our work, the special thing is about the location of TLS. Actually, I scored the TIL as a stromal TIL according to the guideline. And other things are we made like mature TLS and immature TLS located in the intratumor, peritumor, and non-tumor area. It's because we only review the pre-existed H&E images of the TCGA dataset. So actually, if there is no germinal center, then I couldn't say it's not a TIL, so a TLS. So I separate them into the TLS with germinal center and some lymphoid aggregate with high endothelial venule because I'm a pathologist. So I counted them with the size criteria like 100 micrometer aggregate with the plump endothelial venules with a small, highly endothelial venule associated lymphoid aggregate and TLS. And we counted and scored the number of location of them. And we found that the intratumorally located, the lymphoid aggregate or TLS is most prognostic in every subtype.
So was that all done from resection specimens, or how do you deal with it?
Yeah, all is from resection.
resection
Yeah, treatment-naive resection of the TCGA dataset.
Okay, we're just about done, and the session is about to begin downstairs, so thank you very much. Let's thank the speakers, and thank you all for arriving. Thank you.